Hello, everybody, and welcome to the Human Cell Atlas Developmental Neuroscience Seminar. I'll be your host with Deanne Taylor and Bruce Arno, and today we've got Freda Miller and Sten Linnarsson, and we're honored to have panelists Suzana Herculano-Houzel and Gordon Fishell, who will join us at the end. And we'll kick off now with Sten Linnarsson's seminar. Sten really needs no big introduction. He's known globally as a methods developer par excellence and has applied his innovative single-cell genomics, imaging, and computational analysis methods to the developing nervous system for many years. He's been focusing on mouse, but also in vitro systems and human, and has made seminal contributions both from a technology point of view and in applying the methods to explore the nervous system, different aspects of different elements of the brain, and discovering new cell types along the way aplenty. Sten is a member of EMBO. His work has been recognized by the Eric Fernstrom Prize. We're very proud that he's a founding member and is on the organizing committee and has been a key member of the coordinating committee of the Human Cell Atlas initiative. And he's also a member of the Nobel Assembly at the Karolinska Institute. Without further ado, I will hand over to you, Sten. Thank you.
Thank you. Thanks, Sarah, for the kind introduction. And thanks everybody for joining and listening. I will talk about our recent work applying single-cell analysis methods to basically survey the development of the mouse brain. And this is work that builds on previous work that we published 2 years ago, where we made a large survey of the molecular transcriptomic composition of the mouse nervous system. And in that work, which was led by Amit Zeisel and Hannah Hochgen in my lab with many collaborators, we surveyed more or less the whole nervous system, focusing mostly on the central nervous system, but including the sensory, sympathetic, and enteric nervous systems, and wanted to get sort of a bird's eye view of the molecular architecture of the nervous system. In sort of very high-level summary, what we found was that cell types in the brain could be organized into categories that reflect functional and developmental organization of cell types. And so this plot here shows all of the nearly 300 clusters that we identified in this data set. So each leaf here in the dendrogram is a cluster, so we could call it a cell type, maybe, if we're generous. And they're organized now by, in this dendrogram, by similarity, by transcriptional similarity. So cells that express similar genes are close to each other. Below the dendrogram, you see these bands that show neurotransmitter phenotypes for neurons. So the various blue shades, for example, are excitatory glutamatergic neurons. The reds and the pinks and the oranges are inhibitory GABAergic or glycinergic cell types. Of course, this is all inference based on expression of vesicular transporters and the enzymes to synthesize these neurotransmitters. And at the bottom, you see a band that shows the sort of major developmental compartment that the cell resides in. So if we look at this dendrogram, what you can see is that the major split is between neurons and non-neurons. So all neurons end up on one side and all other cell types end up on the other. And this really is a functional split. It just shows that neurons, when they become neurons, induce and express a very large program of genes that are related to synaptic function and axon guidance and ion channels and electrical transmission and all this. But the next split within neurons is between the CNS neurons and the PNS neurons, and so the peripheral nervous system again separates completely from the central nervous system. And this, we believe, is a developmental split. It corresponds exactly to the distinction between the neural tube and the neural crest. So the PNS differentiates early in development, and there are migratory cells that sort of move out into the body to populate these different compartments of the peripheral nervous system. And all of these cells retain memory of this early developmental event. And the same is true within the CNS. So these subtrees here largely correspond to developmental compartments. So for example, you have forebrain here, you have midbrain over here, hindbrain here, spinal cord. And so cells tend to remember how and where they were born in sort of transcriptional terms. And cells that reside in the same anterior-posterior compartment tend to be similar in gene expression. The exception, curiously, is all the cholinergic and monoaminergic cell types, or almost all of them, which are more similar to each other and form this sort of subcluster here, despite being located in both the forebrain, midbrain, and hindbrain. But to follow up this, then, we did a very systematic survey of mouse brain development. So we're focusing on brain here only. And we sampled every day of development from E7, which is very early. It's sort of the neural tube stage, and until birth. And we, in the beginning, sampled large pieces. You can see an image here of the embryo. So either the head or the whole embryo in the beginning. And then as development progressed and the brain matures, we were able to dissect into finer subdivisions. So, for example, forebrain, midbrain, hindbrain, or even finer distinctions in the forebrain. Again, this is a survey type experiment. This work was led by Joelle in my lab and Kim Siletti. In distinction to the adult data set, developmental data is sort of another few dimensions of complexity. So in this particular data set, we identified 942 clusters, as you can see there on the right. We divided them into 18 major classes and 51 subclasses. These plots are t-SNE plots that show all the approximately 300,000 cells that we analyzed. And how they are related. And at the bottom, you see a dendrogram of all the cell types. But the problem now is that this is we're merging time, space, lineage, and cellular maturation here. And so it's a really big challenge to visualize this kind of data and to sort of wrap your head around it. If we start with a simple visualization, if we take those major classes only, we can get this sort of quantitative view of brain development that really shows all of the major events or waves of differentiation that form the brain. So if we start at E8 here, at this early stage, we have cell types that belong to the early neural tube and to the germ layers, and to the neural crest, or the future neural crest. And we have a few vascular cells as well. And then very rapidly, these are replaced by the growing neural crest and the emergence of radia glia that form the neural tube as it matures eventually into neurons. The radia glia are replaced around E10, 12 by neuroblasts, so immature cells that are about to become neurons, which in turn differentiate into neurons, and eventually you end up with a large abundance of neuronal cell types. Glia are really late in this scheme, so you see oligodendrocyte precursors originate around E13, E14, but they're really a minor population until the end of this timeline. Astrocytes really aren't very prominent here. We see some, actually, I think I'm missing the legend here, but there's a sliver of orange here that represents the very earliest astrocyte types that develop here towards the end of gestational age. And then you have, at the same time, cells that don't really belong to the brain proper, but for example, the meninges in the form of these fibroblasts that develop quite early and envelop the brain, and the microglia that actually develop outside of the brain and invade the brain during early development, and then stick around to populate it and defend it. But of course, this is unsatisfactory. It's a kind of a aggregate view, gives you a high-level view of what's going on. But really, we want to see all of these different phenomena, space, time, differentiation at the same time. So in order to do that, we tried, actually inspired by old maps of the Earth, we tried to use the idea of an actual map. And in actual maps, you will see layers that convey different types of information. And in particular, in these circular maps that were popular a long time ago, leaves space in the corners for sort of miniature maps that show additional information. So to walk you through this complicated figure, first of all, in the middle, you have the t-SNE of all the cells. So this is the same as I showed you before. Now it's colored by these major classes of cells. And to appreciate how this is organized, you'll see that the very immature cell types are down here. So the early germ layers and stem cells and so on. And then you see a slightly more mature cells like radioglia or neural crest in the middle here, and then sort of branching out into, for example, meninges over here, glia here, and then all the different types of neurons over here. Complementing this view, we have around the perimeter here all the 942 clusters that we defined. And they're shown as a dendrogram of clusters, exactly like before, although we've cut it into 25 larger pieces that represent sort of more intermediate subclasses of cells. So for example, ependymal cells here, glioblasts, so cells that are destined to become glia, although they are still very immature, microglia, OPCs, and so on, and all the neurons over here. This is also colored here in these bands in the same way as here. So you see the green band here, for example, corresponds to all the neurons. Now, to orient yourselves, we can plot markers. So this is a plot of major markers of cell types. So for instance, you have SOX10 here marking the oligodendrocyte lineage, OPCs. You have AQP4 marking glioblasts and future astrocytes, AIF1 marking microglia, and so on. So that gives you a high-level view of the different categories of cells. We can also plot age. So this is the age of the embryo that each cell comes from. And you can see here, actually, luckily, the trunk of this plot organizes itself quite nicely in time. So again, you have the earliest stages here before E9. And then you have the neuroepithelium and neural tube here differentiating into these different major lineages. We're also interested in space. So we can plot the spatial location from which we obtain the cell here. Now we're plotting just broad categories. But here you can see, for example, that this big branch over here that we've labeled cortical pyramidal includes also hippocampal excitatory cells, by the way, consists solely of forebrain neurons. Whereas in the glia and the meninges lineages, you see a mixture, and there's really no distinction between the forebrain, midbrain, hindbrain in those regions. Although, as we'll see a bit later, there are still, if you zoom in, some differences that can be observed. And finally, we can overlay cell cycle, and showing that really, as soon as cells decide which major lineage to enter, they also exit the cell cycle and start to differentiate. All right, so the first thing that happens in brain development is really the definition of the germ layers and of the nervous system as so sort of laying down the nervous system in the anterior part of the embryo. And here we made a comparison with a previously published paper by Pijuan Sala et al. That looked at the earliest stages of gastrulation and sort of the stages just before our data set begins. And aligning, so in our data set, E7 and E8 comes here. So you have these early stages and the very early neuroepithelium in our map. If we take only these cells, we find that they belong to 3 different categories: the primitive streak, the other lineage progenitors, and the neuroepithelium. And we can label them with markers. And we can also overlay our cells onto this previously published data set and see nicely that the primitive streak stage corresponds to this very early cell types here. And then you have the various non-neuronal lineages branching out here. So the extraembryonic endoderm, for example, and the ectoderm. And then you have the neuroectoderm and the neural crest mapping here in the middle, corresponding to exactly to where it's located in that data set. However, since our data is a little bit later and a little bit more focused on the brain, we have a larger number of neural clusters, where in the reference data set here there was only 5. We have 10 or so. And if we take those particular clusters, so now we're focusing in only on the neuroepithelium, what we find is that these clusters here are really related to forming the rostrocaudal extent and actually the spatial patterning of the very earliest neural tube. So one example of this is this cluster up here, the neuromesodermal progenitors that correspond to neural progenitors at the very posterior tip of the embryo. Stage 2 in making a nervous system is laying down the patterning of the brain. So organizing it into anterior posterior compartments and then that express the known morphogens like FGFs and hedgehog and BMP and Wnt and so on. And in this way, we identified these 10 clusters. And indeed, they express the expected morphogens. So for example, floor plate organizers express sonic hedgehog and so on. But interestingly, we found many more organizers than you would usually find defined in the textbooks, and they express other morphogens and diffusible, well, potential morphogens, I should say, diffusible molecules that distinguish, for example, the floor plate of the midbrain from the floor plate of the hindbrain by diffusion of specific diffusible molecules like BMP7, for example. So what we think is happening here is that the organizers are actually more complex, and there are more sort of subtypes of organizers that are active in different parts of the brain, and they are secreting many more secreted ligands than the classical ones. To validate this, we teamed up with Mats Nilsson's group and performed in situ sequencing. So these are sagittal sections of an E9, E10 embryo. And first, to just validate the classical location of the organizers, we stained with these, for example, sonic hedgehog for the floor plate, and you can see that you have the midbrain floor plate here, you have forebrain floor plate here, and the hindbrain floor plate here. But then adding now genes that distinguish particular segments of, for instance, the floor plate, we could define, for example, here the floor plate of the midbrain by the expression of NTN1. Or the floor plate, the roof plate of the midbrain using a combination of TAL2 and Wnt8b. All right. One final thing is, given all this data, what you really want to do when you have developmental data is you want to understand how the cells are related by lineage relationships. So how would they potentially differentiate? And this, of course, can only be inferred from this kind of data. We cannot directly measure it because this is snapshot data. But so our attempt to infer it was to construct what we call pseudo-lineage trees, where in essence, we take each cell and we define a small neighborhood using information theoretic measures. So a neighborhood that contains cells that are near indistinguishable in an information theoretic sense. And then we link cells that fall within each other's neighborhoods. And doing this, you create a graph. Now, if you designate a particular root cell, so we designated arbitrarily a root cell here in the neuroepithelium, the earliest neuroepithelium, then if you take now a path from this root cell to any other cell, you will define a tree. So there will be branch points and there will be leaves or tip cells that correspond to the ends of this tree. And importantly, because we're applying a strict radius, we cannot move outside of the neuroepithelium here, for example. So we can connect this root cell to the meninges, for example, makes sense through the neural crest, but it's not connected to the endothelial cells because there is just no way to get from this cell to completely unrelated cell types. Here you see an example now of 10 randomly selected lineages, and you can see how they connect the early neuroepithelial cell, for example, here through radioglia to glioblasts to an early astrocyte, or here through a forebrain radioglia through intermediate neuroblast stages into a upper layer cortical neuron. We can also apply these pseudo-lineage trees by cluster. So we can take all the pseudo-lineages that end in a particular cluster and we can trace only those. So for instance, here you have all the cells that we defined as cortical excitatory neurons, and we just trace those back to the same single tip cell. And this gives you kind of a lineage bundle, and you can flatten this lineage bundle, and now you can ask what genes are expressed along this bundle of lineages. And this gives you, so then we use an sort of a statistical enrichment criterion. You can find genes that are expressed, for example, in cells that are destined to become cortical excitatory neurons, but the genes are expressed in the early sort of E9, E12 stage. I emphasize that this is a way of generating hypotheses. Of course, we're not measuring the lineages directly here, but they seem to work pretty well. And we can validate this particularly for the cortical excitatory lineage. Using these genes, we can go to the tissue and we can stain it. And indeed, for example, EMX1 is expressed around E11, and it's expressed here in what will become the cortex. Whereas, for example, SATB2 appears around E15 and then stays on, and becomes in late stages the upper layers here of the cortex. So with that, I'm ending, and I want to point out that we share all this data. We have a website at mousebrain.org where you can browse and download all of this data, both for the adolescent and the developmental atlases. We share all the raw sequence data and also all the annotated data and so on. We have a nice interactive version of the atlas plot where you can click on things and interactively plot genes and so on. I should say I checked it just before my talk here. The front page seems a little broken, so we're going to work on that to get it up and running again. But I think if you just click past the front page, it's still operational today. So have fun with that. And with that, I want to also thank all of the people in my group, all our great collaborators and funders, and I'm ready to take any questions in the panel discussion.
Clap, clap, clap. Thank you, Sten. Fantastic talk. And we look forward to picking up in the panel session later. So it's a tremendous pleasure to introduce Freda Miller. Freda is a cellular and molecular developmental neurobiologist, and she's based in the Hospital for Sick Children at the University of Toronto. She's also a Howard Hughes Medical Institute International Research Scholar and a Canada Research Chair. a fellow of the Royal Society of Canada, and a fellow of the American Association for the Advancement of Science. Freda works on mechanisms of regulation from stem cells to their lineages and development, focusing on both dermal and neural stem cells, I believe, with a particular focus on the nervous system. And we look forward to hearing more on that topic from you now, Freda. Thank you for joining us.
Great, thank you very much, Sarah. I'd like to start by actually thanking, let me make sure I share the right thing. Thanking the organizers for giving me an opportunity to participate in this forum. One of the main interests of my laboratory, as Sarah indicated to you, is how you can go from a small population of proliferating precursors, which you've just heard about from Sten Linnarsson, to build a nervous system that allows me to give you this talk today. And in that regard, what I'm specifically going to talk to you about is the attempts we've been making to use single cell approaches to try to understand that question. Now, if you want to ask a question like how stem cells build a tissue, you really need a model system. And the model system that we've chosen to focus on is the developing mouse cerebral cortex. And while there are many reasons for that choice, for example, the cortex is the site of most of our higher cognitive functions, as a molecular cell biologist, the real reason for that choice for us is the fact that the precursors that build the cortex generate cells in a highly stereotypic fashion. We've chosen the cerebral cortex of the mouse as a model system because it generates neurons and glia in a highly stereotypic fashion in time and space. So if you actually look at the developing cerebral cortex at around, in the time frames that you've just been hearing about from Sten, at around E10 or 11 or so in the mouse, it's just basically proliferating precursors. And then around embryonic day 11 or 12, these precursors, which I will call radial precursors, others call them radial glia, start to generate neurons, and they generate throughout the entirety of that gestational period. They generate the various different kinds of large excitatory neurons that build the circuits that we need. Then around birth, by mechanisms we don't understand, they switch to making glial cells, but it's not over then, because by mechanisms, again, that we don't really understand, a subpopulation of these cortical radial precursors somehow make the decision to become dormant or quiescent and to populate one of two major adult neural stem cell niches in our brains. And in this case, this is the forebrain neural stem cell niche that is right around the lateral ventricles. And somewhat surprisingly, the cortical precursors that persist to do that now make a completely different kind of neuron. They make GABAergic interneurons. Well, several years ago, when the first DropSeq paper from Evan McCosker and Steve McCarroll was published, my laboratory became very excited and said, wouldn't this be a fantastic way to understand all of the different cells that are in the cortex while these events are happening and to really understand their phenotypes? And with this in mind, we did an intellectually very straightforward experiment. We simply dissected out the cortex over this entire period of neurogenesis, and we used DropSeq to sequence a bunch of cells, and we asked what we saw. And basically, when I say we, I mean Scott Uswa, a postdoc who now has his own lab at University of Toronto, a wonderful graduate student, Michael Borat, who's just finishing now, together with an equally wonderful graduate student from Gary Bader's lab, Brendan Innes. And while I don't want to go into all of the conclusions of that paper because of time constraints, I would like to introduce you to two of them. One of the first conclusions, which was a bit of a surprise to us, is that in spite of the fact that these radial precursor cells were making diverse types of excitatory neuron progeny throughout this time frame, that they themselves at the transcriptional level stayed relatively homogeneous and did not change. And in fact, we were able to take advantage of that homogeneity to define a transcriptional core signature of about 90 genes that were highly enriched in those particular cells. But the second bigger surprise came when we compared that data to data that had been published from Fiona Deutsch's lab, microarray data, in this Kodega et al. Paper, where they had basically isolated what they thought were quiescent adult neural stem cells from the forebrain niche and simply asked what they were enriched for. and much of our gene signature was still enriched in these adult neural stem cells. And basically what that led us to think is that in spite of the fact that I think most of us think that the cells that build tissues during embryogenesis are distinct from their adult stem cell progeny, that this may not be the case. That in fact, these may be the same precursor cells that have simply persisted in a different activation state and in a different niche. Well, in order to ask that question, we chose to basically follow these precursor cells throughout their lifespan. But there was a complication to this analysis, and I'm going to walk you through that here. Basically, if you look at this schematic on the bottom here of what this forebrain adult neural stem cell niche looks like, this is the lateral ventricle here, and the stem cells are scattered around it. What you see is that those stem cells come from two distinct developmental origins. One of them are the cortical cells I've been telling you about that live here and here, but the majority, about 60% of these adult neural stem cells, actually come from a completely different place in the brain. And now if you look at this schematic of the embryonic brain up here, what you see is this is where these cortical radial precursors are, and immediately adjacent to them is a region called the ganglionic eminences, which contains another population of radial precursors that comprise the rest of these adult neural stem cells. And importantly for this talk, the developmental history of these two types of stem cells is somewhat distinct. As I told you, the cortical ones make these big glutamatergic projection neurons and then switch by mechanisms we don't understand to making these GABAergic interneurons postnatally, whereas the GE-derived ones make GABAergic neurons throughout their entire life. Well, because we had this additional complexity, we thought that if we really wanted to definitively do this, we needed to be able to identify cortex versus GE-derived cells. And to do that, we combined our single-cell RNA sequencing analyses with lineage tracing and taking advantage of some really nice mice that have been previously analyzed in detail. And when I say we in this context, I mean Michael again, I mean Daniel Jiang, a terrific MD PhD student in the lab, and Neri Tamasian, another younger student in the lab. And basically what they simply did is they went from embryonic day 14 through to postnatal day 60, focusing on this region of the embryo and the adult, and sequenced a bunch of cells to test our hypothesis. And now I'd just like to very briefly walk you through some of the conclusions of that analysis. So the first thing came from the comparison of the cells that build the cortex versus those that build the ganglionic eminence. They do completely different things embryonically. Nonetheless, in spite of that, they are highly similar at the transcriptional level. And I just want to walk you through an example of the data that allows me to make that conclusion. This is simply a t-SNE of the embryonic data that we gathered from our lineage tracing mice. This is a gene expression overlay to show you the cortical lineage tracer, and you can see we have nice segregation of cortical and GE-derived cells. We then extracted the radial precursors from that and then directly compared radial precursors of two different origins. Now, if you do that, one of the things that you see is that when you do, for example, a Pearson correlation of average gene expression, is that they really do look very similar. So this is an R value of 0.99. So, and you can see that just visually here. But nonetheless, I don't want to mislead you. There were a handful of highly distinct genes that obviously must be involved in specifying the functional differences between these cells. And we were able to identify those genes, and I can tell you that many of them have previously been known to be important for making excitatory versus GABAergic neurons. We saw this same profile throughout life. So what happened then when we looked over life and compared the embryonic stem cells and their adult neural stem cell progeny? Well, in order to do that, we simply extracted out the various precursor populations at all the time points that we analyzed. And we regressed out the cell cycle to make sure that we weren't just looking at things that were driven by proliferation. And then we did a very simple monocle trajectory analysis. And basically, I think the conclusion is pretty clear here that it's a really simple trajectory, that we have the embryonic radial precursors at this end, we have the adult neural stem cells from postnatal day 20 to 60 at this end, and then we have this gradual transcriptional transition from one to the other. And I can tell you that we did a lot of analysis asking what this transition was, and that it doesn't appear to be a change in identity. Instead, it appears simply to be a shutting down of all transcriptional programs associated with an active versus a dormant cell state. So again, this made us take a step back and say, well, if that's really true, that it really is a similar cell or very similar cell, except in a different activation state, then you might predict that when you go backwards, when you reactivate these cells to make adult-born neurons, perhaps they'll go back to this earlier embryonic state. Now, for you to understand how we ask that question, you need to understand the lineage in the adult. So we have dormant neural stem cells. I've been telling you about that. They become activated by mechanisms yet unknown. And then they generate transit amplifying cells, some of which are proliferative and some of which are not. And then those ultimately make the adult-born neurons. So basically, we simply looked in our data set, and now we incorporated these various activation stages in our analyses. And I think the take-home message is very easy to see on this trajectory. This is now including the activated neural stem cells, which, by the way, are not proliferative. And basically, here's the same E14 at one end, adult at the other. And now the activated adult neural stem cells are segregating very closely with those early transition stem cells that are going from being an embryonic radial precursor to a postnatal stem cell. We did a number of different analyses to confirm that this was actually the case. This is a two-dimensional single cell correlation analysis where we've correlated each of the single cells, their transcriptomes, with four different founder populations: cortical RPs embryonic, GERPs embryonic, total RPs, and then along this axis are the embryonic RPs versus adult dormant neural stem cells. These green ones are the E14 cortex radial precursors. The blues are the GE. These are the adult dormant neural stem cells in this plot. And most importantly, these are these activated neural stem cells here in the middle. As you can see, you can almost imagine they're going backwards, although that's of course not literally what this is. But the really important thing I wanted to highlight here is that these purple cells are actually these adult transit amplifying cells, and they're overlaid almost precisely on the embryonic radial precursors. So what I've told you then is that we think that you have a population of forebrain. In this case, we've studied the cortex and GE precursor cells, which of course are to some degree specified by their position, and thus the types of neurons that they make are functionally distinct. But we think that together these populations undergo a dormancy transition to become adult neural stem cells, and that when they're activated in their niche, they essentially just reverse that direction and go back to this developmental state to make adult-born neurons. Now, this, like any good study, I always tell my students, this leads to more questions than it answers. And so I'd just like to spend the last few minutes telling you some of the things that we're trying to answer now based on these findings. So remember that what I've told you is that we have a population of precursor cells which persists throughout life, but that does very different things at very different times. So the question then becomes, what is driving those different cell fates? And this is really a microenvironment question. What are the things in those microenvironments that tell you to do big excitatory neurons versus GABAergic interneurons? Well, we've been interested in this microenvironment question for a long time. And about 5 years ago, we made our first forays into trying to use systems biology to ask these questions. And basically at that point, we sorted out embryonic cortical precursors and their newborn neuronal progeny. And we basically just used cell surface mass spectrometry to define all of the receptors for ligands that were on their surfaces. And we combined that data with bulk transcriptome at the time. And using a ligand receptor database generated by Gary Bader's lab, and then in collaboration with Peter Zanstra, we computationally modeled paracrine and autocrine interactions between these two populations of cells. Now, this was actually a very productive endeavor using this approach. We were able to define a number of new ligands that had not previously been considered within this perspective, that were very important for regulating the genesis of neurons, and in our case, oligodendrocytes in this system. And that was work done by Scott Yuzwa again, as well as Anastasia Voronova, who now has her own lab at University of Alberta. But it doesn't escape this audience in particular that wouldn't this be much better if you could do the same kind of thing with single cell RNA sequencing data? And indeed, we're now doing that. And I just wanted to introduce you. This is actually our first effort, which was just published this year. This is not the brain. This is instead, we asked about the injured nerve and about ligands that were made by various cells in the injured nerve that might act either on the neurons that project in those nerves or on their target tissues. Nonetheless, of most relevance to this talk is that Dina McCrae, a really wonderful research associate in the lab, is now doing a similar kind of modeling, asking about the interactions at a growth factor ligand level between these different cells in the niche and both the neural precursor cells and, for example, their transit amplifying cell progeny. The second question, and this is one we were only able to ask thanks to a really lovely paper published by my co-speaker here, Sten Linnarsson, is the other population of adult neural stem cells in the brain lives in the hippocampus. And what people don't appreciate, I think, is that embryonically, those hippocampal neural stem cells come from an area that is very close to these cortex and GE cells I've been telling you about. So here's cortex. This is a schematic of the embryonic forebrain. And those neuroepithelial cells that will ultimately make these dentate granule neural stem cells are right here. So thanks to this beautiful paper that published from Sten's lab from Hannah Hochgerner, we were able to basically directly compare our forebrain, cortex and GE, neural precursors to the dentate gyrus neural precursors because they analyzed them all the way from embryogenesis to adulthood. And I probably wouldn't be telling you this if they were not surprisingly similar. And perhaps Sten is giving you a hint of this when he shows all of his radial glia being together throughout development. But this really speaks to the fact that, and I just want to show you one piece of data here. I've put up a couple of them here, but this is just a UMAP of the combined neural precursor cells from both Sten's paper and from our data. And I just want to orient you in this case, all of the purple cells would be cortex or GE derived, and all of the green would be the dentate gyrus ones. And you can see here the embryonic ones cluster nicely, the early postnatal ones cluster nicely, and the adult ones come. So they're even undergoing what looks like the same developmental trajectory. And that is something we're analyzing now. And the final question that we're asking, obviously of most relevance to this group, is does this have anything to do with human biology at all? And this is a particularly relevant question, I think, in the forebrain and the cortex, since the cortex is one of the structures that has evolved massively in a human as compared to a mouse. And so again, we were able to ask this question, and we're just starting it now. I'm taking advantage of another lovely data set that was published from Dan Geschwin's lab last year, where they have basically profiled the mid-gestation human neocortex. And Michael Borat has taken this data. He's reanalyzed it through a pipeline that Gary Bader's lab, largely Brendan Innes, has generated. And what you're looking at here is all of that data analyzed in this other pipeline, which isn't that different from theirs. But nonetheless, the point is, I want to point out to you that all of the radial glia are up here. These would be the non-proliferative, and here's the proliferative. For the aficionados in the audience, I want to point out that you can see the outer radial glia and the ventricular radial glia here closely associated, but somewhat distinct from each other. But what I really wanted to point out to you is that we took the core signature that we had defined from our murine radial precursor cells, and we overlaid it on this human data set. And what you can see is that at a gene expression transcriptional level, that core signature identified those cells very effectively. And we're now asking precisely how similar or different these two species are with regards to these transcriptional programs. So I'm going to end there. I think my time must be almost up, and just thank everyone. Much of this work has been done in collaboration with my longtime collaborator, David Kaplan. I think I've mentioned everybody who actually did the work, but obviously this whole initiative in my laboratory went forward because I had some amazing collaborators. And in particular, I want to highlight Brendan Innes in Gary Bader's lab, and thank all of you for your attention.
Thank you, Freda. The fantastic talk. Good. I think we can now move to the next section of this little symposium. And Suzana and Gordon are going to join us for this part. Suzana Herculano-Houzel is, I would say, an evolutionary biologist and neuroscientist at Vanderbilt University, and Gordon Fishell is a neuroscience professor at Harvard. And so welcome both of you. Thank you for joining us. We're very honored to have you. And so together with Sten and Freda, we'll now move over to the Q&A and discussion part and have a very open format here. We also have the other Human Cell Atlas development panelists here in this symposium who may also want to chime in. So going over to Slido and kind of kicking off there, maybe a nice topic to start us off is whether there's one question on from Bernd Jagla, whether there are pathways that are developed in parallel on different cell types. And you know, one of the things there's sort of patterns that were similar across Sten and Freda's seminars with respect to cell cycle, signaling circuitry, and so on. So I wonder whether each of you would like to comment on that.
Can I put a little context to that question?
Sure.
One of the things both of you, Freda and Sten, mentioned are signaling factors. You put it, Freda, in the context of growth factors. Sten, you mentioned morphogens. Can you talk about how you think the intersection of those lineage tracks are affected by morphogens, and what makes a lineage track accessible to a morphogen at one point and inaccessible at another, because clearly those morphogens or signaling molecules are used pleiotropically.
Do you want to start, Sten?
Yeah, right. So I think this question is really good, and let me maybe reinterpret it. And so the question is about whether there are shared gene modules that are reused between cell types, or whether there are sometimes analogous or paralogous gene modules that are used between different cell types in different parts of the brain. And then specifically in the context of signaling molecules, you know, there's a similar question for those. And I would say I'm extremely frustrated because working with this kind of data for a long time, I get the feeling that there is a relatively simple organization here. It's just slightly too complex for my brain to wrap around. And I think you could see from Freda's work as well that there are these patterns that are reused all the time. Things are not as different as we sometimes think. When we analyze everything in terms of cell types, that might not be the correct unit of information or unit of analysis, because inside of those cell types, when we look at gene expression patterns, there's so much reuse and so many similar patterns that come back all the time. And specifically in terms of signaling, obviously this same morphogens are reused. Cells respond in similar ways, but not exactly similar. And I just don't understand the code here. I'm sure there is a code, but I just don't understand it yet. So I think that's a really fantastic you know, avenue for research.
I think Suzana would like to say something next. Is that okay, Freda? And then we'll go over to you. Yeah.
Thanks. Yeah, let me just take that cue and ask you guys about something I noticed, Stan, in the gorgeous plots that you were showing. If we try to put the inferred pathways, the inferred paths that you're reconstructing with what we believe we know from biology, there are two things that caught my eye. One is you have the Schwann cells clustered together with the glioblasts and not neurocrest. And second, you have the oligodendrocyte precursors clustered also up there and not down here with what they supposedly come from, which is something along the lines of what will be the inhibitory neurons. So I was puzzled about that. And then I saw Freda talk, and I'm wondering if could that be the case that you're mapping them at a point where these paths have just reverted back to a state and then gone off the other, or what other possibilities do you could explain that?
Yeah, okay, let me just quickly answer that, and then we'll go to Freda. So for the Schwann cells, it's true that in the plots, they look like they are very close to the late glioblasts, late radial glia. But we looked at that very carefully, and there are some papers that suggest that, at least in injury models, that you can make Schwann cells from radial glia, but there is actually a gap there, and we cannot connect them. So our pseudolineage analysis does not really connect them. They just happen to end up there in the visualizations.
Okay.
The OPCs, however, I think
Sten,
are...
you showed the cell cycle score. I mean, I can't remember the details, but might that be like a confounder here in this scenario?
It's always a confounder. We try to block it by masking all of those genes, but of course, it's still always confounding. I think in this case, it's actually that Schwann cells do share quite a lot of so I think that goes back to the question of modularity, that there are shared features of Schwann cells with, actually I would put it the other way around, that OPCs share a lot with neural crest. So there's a whole gene program that seems to be activated in OPCs that's very similar to one that's activated in neural crest, even though, of course, that's completely different lineages. And for OPCs, I would say that they are in the right place. And we showed in the adolescent paper that when a cell becomes an OPC, it kind of erases its memory of its spatial origin to a very large extent. So that lineage kind of forgets where it came from. Not sure if that's true for the Schwann cells, but.
Can I try rephrasing the question a little bit more? I think this is an absolutely fundamental set of questions. So when you think about what we know from genetics, slit robo versus afferents versus transcription factors, differential transcription factor utilization versus things like in this fractal kind of implication that Freda was talking about. How do you think about those kinds of pathways in like structural differentiation regulation versus lineage, sublineage differentiation?
Yeah.
Freda, they want to?
Sure. I'm going to answer two questions. I'm going to answer first the question of complexity, which is something we think about a lot. How is it that a cell, whether it be a precursor or a neuron or an OPC for that matter, when it's confronted with an environment with many different factors in it, how does it sort that out to do the appropriate thing at the appropriate time? And I think that went back to what Gord was asking. It's been our hypothesis, which isn't strictly true, that you can perhaps get insights into that by focusing on the receptors. So, a lot of our work is focused on ligands, but the reality is we do know that receptors turn on and off through the lineages as they turn on and off through different stages of differentiation for neurons, and they're also spatially localized. Now, the complication there, of course, is that any given cell has multiple receptors. So you've narrowed the question down. It's a little less complex, but it's still very complex. And that's where the million-dollar question of, can we actually define the downstream signaling pathways that would allow us to use these kinds of data to predict what would be dominant in a given situation? Because there's clearly dominance of factors you can, as evidenced by a million cell biology experiments. Now, the question of a similar ligands or signaling systems being used in different ways at different stages. For example, for making a commitment to being a neuron versus actually neuronal differentiation. I mean, that again is something I strongly believe, having worked on growth factors my entire life, that nature uses what it can. And if there is a cytokine or a growth factor in the environment that was maybe used at an earlier stage to promote you, to tell you to become a neuron versus an OPC, then perhaps that's still in the environment and that will be reused to dictate, for example, how much your axon extends or whether you live during the period of naturally occurring cell death. So I think, even 5 years ago, before we had approaches like this, 8 years ago, you became very frustrated because it seemed so complex you would never be able to figure it out. And I completely agree with Sten that now we have tons of information, but we don't know what to make of it. But at least we're a step closer to asking the right questions that will let us know what to make and what patterns to follow. So anyway, I think I talked enough answering that question.
So can I chime in? Obviously, you know, ligands and receptors and protein complexes are kind of one of my favorite molecular biology and biophysics topics. And what we've done is, you know, do global surveys of cell surface and secreted ligands and their receptors, and then look for statistical enrichment of specific complementary expression patterns. And what Freda said, so we have a resource called CellB that provides that online, and it was originally very geared towards immunology, but we've now tried to curate and extend it. And a lot of credit goes to Rosa Bento. And what Freda was saying about, but actually I should say that you know, you think that the sort of immunology and development and neuroscience are two separate things, but you know, a lot of the signaling, the molecules used are deployed in these different contexts. So I saw an interferon family factor mentioned in Freda's talk, I think, and so on. Like what you're saying is right. So nature just redeploys, you know, factors wherever it can, and to look for, you know, the pathways that are really active is hard. It's kind of like a telephone, you know, circuitry problem where you've got lots of calls coming in, lots of calls going out, and how are they connected? How are the signals coming in connected to the transcriptional modules that go out? It's a many-to-many relationship. So deconvoluting that is really complicated, but there are, you know, it is worth attempting to do that. And there are frameworks like Dorothea from Julio Saez Rodriguez groups and so on that attempt to do that, and where you can extract meaningful signals even from all of that complexity. So it is, you know, this type of data, especially combined with spatial information, although the signals can travel obviously and diffuse if they're secreted, it is, you know, I think it will give us more insight, and it will get us towards what Sten was saying about principles and patterns eventually. I'm optimistic. That's my time worth.
Let me ask a question to Sten and Freda about not the differences, but the similarities across. That was a big part of your talk, Freda, as well as yours, Sten. Is there a way of quantitating shared transcriptional pathways in the two, where you can, because the difference is when you do this TSNE or UMAP mapping, you're really just focusing on the genes that are different, and the great majority are the same. So I wonder how much insight you can get by, instead of looking at genes individually, to really structure it into signaling, internal extrinsic signaling pathways, and really ask how much shared utilization there is among them.
That's a great question, Gord. And I think that we have not, I mean, it's a really interesting way to look at the data, and we have not done that. You know, we did try to, one of the reasons we did a lot of correlations, including the single cells, was so that we weren't just looking at the genes that were differentially expressed, right? We really wanted to look at the whole transcriptome. But I had not considered actually using the receptor ligand information as a way to ask these questions in a broad and perhaps to ask about the questions we've been talking about actually ever since the discussion started. How similar are these pathways between different developing cells, and are they used at the same stage? I think that's a very clever idea. And if you did have, from the kinds of things Sarah's talking about, you know, of course we don't have transcriptional readouts for all the pathways, but we do for some of them that are very hardcore transcriptional readouts. If we could somehow combine that information and use those as modules to interrogate these big data sets, that would be a really interesting thing. Sten, have you done anything like that?
So we try, I mean, I was very optimistic a few years ago that we could sort of decompose the transcriptome into modules, and we could find these modules that would be reused, but it's not that simple. Actually, there is there is a small number of modules like that. Cell cycle is the most prominent. I think cell cycle by and large is the same, at least in in all the different brain regions that we've studied. Cells that cycle, they use the same genes. It's not, and it's really binary. It's either on or off. It's really cells are in the cell cycle or not. But that's very unusual, and for the rest, we haven't been able to find clearly, clearly distinct modules like that. You can go in with prior knowledge. So for neurotransmitters, we were quite happy with. We think we were able to sort of infer the neurotransmitter phenotype using just a small handful of enzymes and vesicular transporters. But I wouldn't say that we could discover that from the data, really. But I mean, that may be fine as well.
Yeah, so there are some known kind of transcription factors that link to signaling pathways, and then there's some that can be inferred, but it's a telephone exchange kind of complexity,
I think it is very modular, and there is a system to it, and the system is not too complicated, but it's just a little bit more complicated than we are able to resolve at the moment. It's not just simply modules that are
the system. So in the endometrium and the menstrual cycle, we have some beautiful modules of signaling. But anyway, I want to go back to Slido. I want to go to the audience questions, because that's why we have this amazing online system. And I'd like to go to a question from Bruce, and I don't know which Bruce this is because there's no last name, but the question is, in trying to distinguish lineage, sublineage programming versus migratory structural differentiation mechanisms, how much of it is hardwired versus how much is locally adaptive? So what's dependent on the regional context? What are the major regulators of migration and determination of structural subtypes? So I guess in a nutshell, it's basically what's cell intrinsic and what's from the context. Over to the panel. I know that Suzana was wanting to answer. It's open to anybody to answer, please.
I had another question to ask that was related to the modules, but I'll wait.
I'll take on the environment one. I mean, now you're going to get a very biased perspective, because I kind of believe that environment is at least half the story. And I just want to go back to this cortex versus GE example as an example. So I told you that actually the precursors that build the regions that come from the GE, like the striatum, and those that build the cortex look very similar. What I didn't, and yet they make completely different kinds of neurons. What I didn't tell you is that there's a lot of data out there from various types of studies, including some old work from Gord Fishell, who may want to pipe in on this, arguing that actually that's environmentally determined. So if I take a cortical precursor and I put it in the GE environment, it switches to making what's appropriate for that location, and vice versa. And there's even some adult neural stem cell papers where people have done things like taken stem cells from the hippocampus and put them into the forebrain and said they made the right kinds of neurons there. So I think there is positional information that is given largely by the microenvironment, and that positional information is really critical for telling you what to functionally do and what kind of neuron to make. And that probably undoubtedly and does involve transcription factors that we know are very important determinants. So then you have, as Sarah was saying, the pathway from the microenvironment and the position down to the transcription factors, and ultimately to the functional output and the morphological differentiation of the appropriate neuron. So that's a very biased perspective. Someone may believe more in sort of hard wiring than I do. So perhaps you should pipe up.
That's the sort of French flag question, the kind of Lewis Walport French flag development. Yeah. Sten, do you want to weigh in on that?
I don't know half as much about this as Freda, but my sort of personal opinion about this would be that I think a lot of the fundamental organization of the nervous system is hardwired at some stage, and that there's also quite a bit of stochastic development, that a particular stem cell in one particular region might generate 5 different types of neurons, in some particular proportions, but that this is not necessarily regulated at all, apart from just the probability distribution of generating each of those 5 types of neurons. We don't need to necessarily posit that there is a regulatory mechanism other than sort of just stochastically generating a panel of neurons. I should say quickly that I don't have any evidence for that. It's just my kind of impression of the possibilities.
Did you want to add something, Gordon?
You know, the experiments Freda's talking about, to put it in context, were ones I did many, many years ago, where what we found was if you took progenitor cells and just introduced them in the lateral ventricle, irrespective of whether you took cortical or subcortical, they could reintegrate across paleo, subpaleo boundaries and transdifferentiate. And the more we've looked at the actual differentiation in kind of in vivo context, the more those results have puzzled me, because they're clearly highly specified from an early state. And what I've ultimately concluded is those experiments are very akin to how you make chimeras, where you take inner mast cells and they can reincorporate. So I think there's something very special about the earliest progenitors. And when you do that kind of experiment, you select from progenitors that have no intrinsic specification. So one of the things I'd be really curious about is if you could find those cells that had the capacity to do that and ask what is really changed between them and ones that can't do that. I think there's really space for some experimental biology that you could do where you challenge the cells and then query their state. I really think if someone started diving in like that, we could learn a lot.
I want to put a question out there for Sten and Freda that I just want to have you have the chance to answer. There was one very simple question, which is, what have you learned compared to the previous literature? Like, what are the biggest novel discoveries? So if you could just, I think people got a bit information overloaded, so please.
Yeah, well, I can start. So if I can sort of take these 2 papers together, that one was for the adolescent nervous system and the other was for the development of the brain. I think the biggest surprise for us was that we discovered a very clear set of distinct astrocytes. So astrocytes have been, you know, there are different subtypes of astrocytes that have been known for a long time, but these that we found were kind of orthogonal to the classical one. Protoplasmic and fibrous is sort of the division that's been used in most of the brain. Then there are specialized type in the retina and in the cerebellum. But we found that really the telencephalic astrocytes are very distinct from everything behind the telencephalon. So from the diencephalon, midbrain, and hindbrain is one type, and telencephalon is another type. And then there were some additional minor types in, for example, in the olfactory bulb. And what's surprising about that is both that, of course, that it's not been described before, but also that the border between these populations when we did stainings was super sharp. It's really a sharp line through the brain that distinguishes these 2 major types of astrocytes, and it goes exactly between the telencephalon and the diencephalon. In the developmental work, we went up to E18, so it's just the time point where astrocytes start to develop, and it's kind of borderline that we even get them. But we clearly do see immature cells that are most likely going to be astrocytes, and there are kind of 2 paths that generate 2 different types, and they correspond, I think, to the telencephalic type and the non-telencephalic type. Although we don't have enough cells and we don't have them quite mature enough to really make the connection. But I think that in my mind shows or suggests very clearly that these are developmentally specified types that have a regional extent, and they are really territorial. They don't migrate around, and they're not really induced by the environment. They really are specified from development. That's how they look to me, and I think that's a quite a surprising finding.
So I have to say that from a developmental biologist perspective, the single cell RNA sequencing approaches and single cell approaches in general are, I mean, really, I view it as a revolution in how we dissect these systems. I mean, just to put it into perspective, before we had these kinds of data, we had to, immunostain for 2 markers or 3 markers or something, and like do, inject the mothers with BRDU or something to look at cell cycle status. So just as a tool, it's been phenomenal. But in terms of messages, I mean, you could highlight messages at every level if you're interested in how cells decide to be neurons, for example. One of the things we found, which I didn't emphasize at all, is that many of these precursors are transcriptionally primed to make multiple types of progeny. And that post-transcriptional regulation, and in our case, we focus on translational repression, is a key mechanism for allowing them to make one thing versus the other. And you could say, well, we've seen that in other types of cells, but in the nervous system, because of the complexity of the system, it would have been very difficult to be definitive about that. The second thing, from my perspective, because the other thing my lab is very interested in is trying to recruit adult stem cells to promote endogenous repair of tissues. And so from that perspective, I mean, it's still almost too early days, but if the story that we're building up in our heads about the possibility that these adult neural stem cells really are very similar to their embryonic counterparts is true, then one would predict that the factors we're defining that regulate cell genesis in the embryo may well do so in the adult, and we may actually be able to learn how to expand the potential of the adult cells in order to recruit them towards repair. And we've already had a bit of luck with that approach. So I think that those, I can't say we've identified one new cell type the way that, or several new cell types, I should say, with Sten's beautiful work. For me, it's been almost more an opening up of the world that we can ask about.
Freda, has anyone, or perhaps have you, tried a more directed attempt of finding a Yamanaka-like factor for adult stem cells? I think the answer is that the adult stem cell in its natural state has quite restricted potential.
Yes.
So the question is there a way to expose it to something like a Yamanaka factor and broaden that?
Yeah, that's an excellent question. And as you know, lots of people have been putting in, ASCL1 and whatever to promote differentiation in vivo, or we've taken a little bit different approach. We've speculated that in the normal environment, they are restricted, but the question then becomes, if we get them out of their normal environment, will they be less restricted? It's kind of what you were talking about before. And so our taking them out of the normal environment is to injure the brain in various ways, and then to profile those same cells in these different states. And we're using both single-cell RNA sequencing and ATAC-seq because we want to look at potential, not just what they're actually expressing. Are the genes for excitatory neurons now open, for example, in the tabs? And, it's really too early to say much about this, but we're getting some very exciting changes, Gord. And so that may give us a Yamanaka factor that is actually an environmental factor. And it won't do everything, but maybe it'll do enough that we can actually use that for something.
Yeah, that's fantastic. Makes a lot of sense. Thanks. I was just wondering whether, Suzana, I cut you off before, so I want you to chime in, and then I'd like to move on to the next question, please, Suzana.
So I wanted to take Sten's cue on astrocytes, and something that especially from my own personal work bias, it strikes me from the beginning of this whole field that you guys have started and are working on, that we consistently have all these neuronal cell types, but very few non-neuronal cell types. And I want to take this opportunity to ask you guys, how much do you think that's actually true, given that Stan was talking about this orthogonal way of talking about astrocytes, and how much is simply because we're neurocentric? Have non-neuronal cell types been looked at enough? And my own particular interest right now is the vasculature in the brain, which from the quantitative point of view seems to be one and the same. Capillaries are capillaries, doesn't matter where you look. So Could you comment some more on that aspect of lack of diversity in non-neuronal cells? Is that real, or is that just an artifact from our skewed interest?
Sten, do you want to try?
Yeah, absolutely. Sorry, my internet got unstable here for a few seconds, but yes, I would say that it's definitely true that non-neuronal cell types are much less diverse in the brain. I don't doubt it. We've done very unbiased, as far as we could, sampling of cells from the brain. In the mouse, we used whole cells, so there could be some bias because of size effects and fragility effects. But if you disregard those for a second, clearly, if you just do an unbiased sampling, you get way more diversity among neurons. For example, in the adolescent data set, we didn't even select against oligodendrocytes. So we had, I think, of 500,000 cells, we had like 200,000 oligodendrocytes in that data set, which is ridiculous. And still, it's just one trajectory, more or less. It's OPCs going through a few stages of maturation. And then there is some kind of spreading out, maybe, in response to local cues. The mature oligodendrocytes are a little bit different. I think that's true, but this is nothing like what you see in the neurons, where there are hundreds and hundreds of cell types. So I think that's true. On the other hand, I would say that the most interesting things we've found have been among the non-neuronal cell types, and that's true for all of the projects that we've done to date. And I think that's precisely because, as you said, the literature is very neuron-centric. In the study of human brain, it's even cortical-centric. So just going to a different part of the brain, you're bound to find interesting things, I think, in the human brain.
Yeah, just to add to specifically your question about things like vasculature and the stromal cells associated with large vessels and things like that. We have been actually, Suzana, starting to compare. I mean, we have a lot of data sets on the nerve, for example, which at least is peripheral. And then we also have them on things like dermis, and I have a whole other life with digit tip regeneration. So completely outside the brain tissues. And I can't say this definitively, but from what I can see, the vasculature cells don't look that different in the different tissues at the transcriptional level, but we have not looked for subtle differences that could be very important.
Thank you, that's fantastic. Could I move on to another question that I think is from Gary Bader? Gary wrote, to completely map how the human brain develops, is it sufficient to map both mouse, for instance, with lineage tracing, like what you were saying, and human, and relate them transcriptionally? Or do we need other models and other approaches? And this is a great question, I think, because it addresses evolutionary questions, it addresses medical questions. What's the utility to medical researchers, animals to human development, and so on. So would people care to comment on that? Maybe Suzana?
Gladly. So I'm openly not a fan of this is mouse and this is human data, and contrast the two when here we have the answer to how humans came up. I think we know enough by now to understand that you need to give, to use one of Gord's favorite words, context. You need to give context to how how human brains evolved and how mouse brains evolved, also because we have this tendency to think that mouse brains got stuck in the past somewhere and our brains are more new and more advanced in some way, and that's absolutely not true. We're just two coexisting species. So I think before we can go there, we will absolutely need to expand this kind of analysis systematically to at least a few other non-human primates and at least a few other rodents, and preferably, I keep insisting on this, we need large-brained animal models for comparison, because there are all the issues that come with simply having more neurons, simply just more of all of this stuff that you're never going to be able to glimpse from just looking at mouse brains that we know have 1,000th the number of neurons that we have in our brain. And
a cell atlas of every living organism on the planet.
No, we don't need to go there. There are patterns that we know already. So we can, I think pigs would be a pretty good model to consider, especially because we know that pigs have as many neurons in the cortex as baboons, which is a lot of neurons. That's about 10 billion neurons. Sorry, not 10, 1 billion neurons. And yet they have different sizes, so that gives you an interesting model to look at. But so no, we don't need to look at every single creature alive. We know enough to be smart about the sampling now. But what I wanted to say previously, going back to the idea that there are modules, that there might be gene modules that lead to blocks or structures or chunks of cell types that develop in a certain way and maybe even get connected together. That's an idea that I think that deserves some careful looking at using these methods, especially in arthropods. So maybe fly brains, because we know that evolutionary, if you stick to the developmental definition of homology, we know very clearly that vertebrate brains are not homologous to arthropod brains. And actually the whole invertebrate brain is a fantasy. They're incredibly diverse. I call them non-vertebrate species simply because invertebrates is not fair.
I mean, there's a Drosophila monogaster cell atlas project.
Right,
which will make you happy.
and yet, if you ask Nick Strausfeld and Frank Hirsch, they'll tell you that you do find in the arthropod brain, even though it's not homologous to, say, a mouse or a human brain, you do find these suites of genes that seem to define excitatory and inhibitory circuits, really, not even just simple cell types, but circuits, how they connect to each other. So I wonder, it seems to me like there's a very ripe field for the taking out there, especially if we're interested in these modules. So I wondered if either of you guys are familiar with the kinds of things that we usually don't look at beyond mice and humans, especially in the arthropod world.
I think she's asking,
has there been has anybody looked at transcriptomes and cell specific and in cell type specific diversity in arthropods, say, that you know of? And how does that compare to
Platanarius? So Detlef Arendt likes that organism, which is a very
octopuses,
or
even Drosophila, just plain Drosophila. What do we know? How do
cell atlas project? And I think it would be super exciting and super interesting to compare, like you said, cell types, circuitry, modules, and so on in terms of the evolutionary patterns. I mean, the brain isn't my expertise, but in general, I think that's. Would anyone like to comment on this question about cross-species comparisons from the medical, clinical kind of applications point of view?
Well, I'm sure Sten has something to say. I'd be happy to say something about that.
Please.
I mean,
yeah.
Obviously, I've lived a large part of my scientific life at a children's hospital. So people are very concerned about whether the kinds of things we study can actually be used to either understand or even potentially treat children's brain conditions. And I think that, we ourselves, and others are doing this much better than us, will frequently try to use human cells when possible to try to get the next step along a translational, pipeline. And that can even be something as simple as just some pluripotent stem cell derived cortical precursors, because in our case, we are working on the cortex, right? And, but obviously the organoids is the next step, and there's wonderful people, all over the place doing wonderful organoid work. The problem becomes when we want to go to adults. And so, I can make things that really look right, like developing cortical human neurons in a dish. But What am I going to do when it starts to be an issue of, injury in the adult human brain or a degenerative disorder? And Sten, I know, you are also doing some human stuff. What are your thoughts on this? How do we tackle that?
I think, in one way, single cell analysis and single cell biology has made it possible to use humans as its own model organism in a way, because we can derive so much information from precious tissue. So things that were almost impossible in the past are now doable, because we can understand both the composition of a tissue and also what's going on with that tissue if cells are reacting. We can see, for example, in the tumor sample, we can see the normal cells, the infiltrating immune cells, and the tumor cells, and what they're up to, and we get so much information from even a few human samples. I think that's really, as you said before, it opens up huge possibilities, and it is kind of a revolution.
As a model. Yeah, no, I really like it. Okay, we have one last technical question for Sten that has 11 likes, so I feel I just need to ask it. And that question is, how does pseudo-lineage tree inference differ from classical trajectory inference? And it's by Malte Lucken.
Classical. So I think there's about 50, at least 50 that I know of, trajectory inference methods. And I don't claim any particular huge benefits of this one over any of the others. It is, they are all variations on the theme of linking cells that are similar to each other. And you can do this in many, many different ways. What I like about what we did here was that we apply a strict threshold on the similarity. So there are cells that cannot be linked because they are too dissimilar to each other. So the method doesn't make large jumps. And this only works if you sample very deeply, because otherwise everything will be just their own little island. So it's a method that really builds on having dense sampling.
Fantastic. Thank you very much for that explanation. That's brilliant. Much appreciated. I would like to finish off and just say a huge thank you to our speakers, to Christine and Freda, to our panelists, Suzana and Gordon, and to the committee for these online seminars for the Human Cells Developmental Biology community. Please, also a special thanks, yes, also to the whole committee that's listed here. To the Human Cells Executive Office members, Christine and Tracy, big thank you, and also John Randall, and also the video productions team, and Scott and Russell. Thank you again, and hope to see you next time in December. We have a nice nuggets coming up related to human development and rare disease. Looking forward to seeing you then. There's another version of this for Asia and Australia. Thank you, bye.